Hello, everybody. Welcome to the Inflammation, Fibrosis, and Cancer Journal Club. This is a space that we created for investigators to present their work in inflammation, fibrosis, and cancer, particularly to increase our knowledge and our understanding on these conditions, aiming to find new ways to treat them or understand the way they may overlap or precede each other. My name is Pernia Marin. I'm a physician and co-founder of this journal club with Dr. Mary Salvatore. I will be your host today since Dr. Salvatore is attending some professional commitments. And I am delighted to present our speaker today. Her name is Sofia Tarchi. She is a fifth year student of MedTech, which is a program offered by Humanitas University and Polytechnic Institute of Milan in Italy. She is allowed to obtain a degree in biomedical engineering in this program, as well as an MD in surgery and medicine. And Sophia's interests revolve around the application of AI in the chest CT-based diagnosis techniques, and also the development of new medical imaging techniques to improve early diagnosis of diseases and also patient outcomes. Welcome, Sophia. It's lovely to have you here today.
Thank you. Thank you. It's nice to be here.
Thank you. And Sophia is going to present her work called Breast Stiffness, Risk Factor for Cancer and the Role of Radiology for Diagnosis. And just so we have everything clear, Dr. Salvatore and I are also co-authors in this article, and that's how we know firsthand that Sophia is a brilliant student and researcher. So please, Sophia, take it away. I'm going to allow you to share your screen.
Perfect. Okay, can you see my screen?
Perfect, thank you.
Great. Okay. Okay, so as the title suggests, today we are going to discuss breast stiffness as a risk factor for cancer and the role of radiology in its detection. So radiographic density of breast tissue can vary significantly due to differences in its major components, stroma, epithelium, and fat. Remarkably, nearly half of U.S. females aged 40 to 74 have dense breast tissue. A multitude of factors influence mammographic density. You can find the most important ones listed on the screen. It's noteworthy that high mammographic density is associated with a 2 to 6-fold increased risk of breast cancer. This underscores the significance of our research and its potential impact on improving breast cancer prevention and early detection. The precise biological mechanisms governing tumor initiation and progression in relation to breast density remain shrouded in uncertainty. However, there's compelling evidence to suggest that certain molecular pathways, which facilitate interactions between epithelial and stromal components, may hold the key. Our presentation has three main aims. First, we aim to describe these molecular mechanisms in detail. Second, we introduce a novel term, i.e. breast compactness. And finally, we propose considering breast stiffness as a promising imaging biomarker in the realm of breast cancer research. So, in case of increased fibrillar collagen linked to mammographic densities, the matrix is stiffened and normal mammary development is disrupted. TGF-beta triggers LOX secretion in mammary fibroblasts through a SMAD-dependent pathway regulating ECM and mammary gland development. LOX enzymes remodel ECM by catalyzing collagen crosslinks, boosting tensile strength, and stiffening the stroma. Slight microenvironment rigidity activates Rho GTPases, especially Rho A, governing ROCK, myosin contractility, and cytoskeletal tension. Elevated ROCK expression increases MLC2 activity, which generates cytoskeletal tension through its own contraction. Cells on non-compliant substrates, such as these, experience Rho-driven contractile forces, intensifying tensions on integrins, triggering downstream signaling pathways. Mechanoresponsive proteins like FAK assemble FA at cell ECM contact points, regulating adhesion, migration, and invasion with FAK as a central regulator. Shear stress and cyclic straining activate SRC family kinases, phosphorylating FAs, including FAK, forming the Rho FA FAK loop. the one you see on screen right now. Finally, FAK phosphorylation reinduces Rho GTPase activity, establishing a positive feedback loop, encouraging additional FA formation. So what happens with this FAK phosphorylation? Well, the answer is fourfold. There are 4 things that can happen due to this FAK phosphorylation. They are altered gene expression, cytoskeletal rearrangement, increased FA dynamism, and increased cell motility. All 4 of these steps help the generation and maintenance of invasive phenotypes. Now we're going to go through each of these 4 consequences of FAK phosphorylation. The first is altered gene expression. So the phosphorylation of FAK's tyrosine 397 residue creates a high affinity binding site for the SH2 domain of SRC family protein tyrosine kinases. Recruited SRC phosphorylates FAK at tyrosine 925, creating a binding site for GRB2 SOS, fully activating FAK and the RAS-MAPK pathway. RAS, a small GTPase protein, binds RAF kinase to promote its conformational change and activation. Activated RAF kinase subsequently phosphorylates and activates ERK kinase. ERK kinase phosphorylates and activates ERK. Activated ERK translocates to the nucleus, where it phosphorylates nuclear transcription factors. Altered gene expression. And finally, fibroblasts undergo upregulated proliferation, increased entry into and progression through their cell cycle, establishment and maintenance of a new invasive phenotype. The second consequence to FAK phosphorylation is cytoskeletal rearrangement. Cytoskeletal rearrangement can start in 2 ways. The first is through the association with GAPs and GEFs. FAK phosphorylates alpha-actinin on tyrosine 12. Actin cytoskeleton is depolarized and actomyosin stress fibers are polymerized. Stress fibers are bundled into lamellopodia, filopodia, podosomes, invatopodia, and membrane ruffles at the cell's leading edge. This leads to cytoskeletal rearrangement. Another way we can reach cytoskeletal rearrangement, always starting from FAK phosphorylation, is through the Rho effector M-dia signaling pathway, which is how FAK mediates the translocation of GM1 to the cell surface. Microtubule plus ends are capped and stabilized at the leading edge to establish and maintain cell polarity. Maintenance of a suitable lipid environment for the stability and of local FA microtubule associations lead to eventually the cytoskeletal rearrangement. Third consequence of the FAK phosphorylation is the increased FA dynamism. So growing microtubules accumulate tracking proteins, EB2, at their plus ends. EB2 interacts with MAP4K4, a microtubule-dependent factor and FA regulator, to facilitate its binding to microtubules. MAP4K4 travels to FA sites. MAP4K4 binds to and phosphorylates IQSEC1, a guanine nucleoside exchange factor for ARF6, and ADP ribosylation factor GTP binding protein. In turn, IQSEC1 interacts and activates ARF6, thus promoting integrin internalization through endocytosis. Finally, while FA is detached from trailing edge, it is reformed in the leading edge, thus establishing its dynamism. And finally, the fourth and last consequence of FAK phosphorylation, which we took into consideration, is the increased cell motility. So FAK phosphorylation activates JNK. JNK upregulates MMP9 gene expression at its promoter's AP1 motif. Targeted degradation of basement membrane is promoted and cell spreading and growth are facilitated. Increased cell motility is sensed by FAs. FAs cause FAK phosphorylation. FAs are remodeled, thus further increasing cell motility and establishing a positive feedback cycle. Good. So in summary, we have explored the complexities surrounding breast density and its connection to breast cancer development. While the exact biological mechanisms remain elusive, the molecular pathways involving epithelial stromal interactions seem to play a significant role. These pathways may contribute to the deposition of collagen, leading to increased matrix stiffness and enhanced cell migration, which support breast density and potentially carcinogenesis. These findings shed light on the intriguing link between breast radiodensity and breast cancer. However, further research is essential to fully understand these processes and develop innovative treatments that target carcinogenesis at various levels. This journey of discovery holds the promise of improving breast health and cancer prevention. So the second aim of our study was to define breast compactness and describe its ability to work as an imaging biomarker for breast stromal stiffness. If this were possible, breast cancer screening would be revolutionized, not only informing who was at the greatest risk for breast cancer, but also the area of the breast that is most vulnerable. To do so, we studied STHMI and micro-CT. STHMI is a medical imaging technique that uses a single ultrasound transducer to both create traditional ultrasound images and measure tissue elasticity based on harmonic motion for improved diagnostic information. Instead, micro-CT is a non-invasive imaging technique that uses X-rays to create detailed three-dimensional images of small objects or specimens at a microscale level. On the left, STHMI demonstrates 3 non-cancerous breast specimens, which on visual inspection could be classified broadly as fat, mixed, or fibrous. And on the right, we have micro-CT, which demonstrates how HU, so Hounsfield units, varies from one area of the breast to another. So we see that areas that contain fat present low HU on micro-CT and have slow STHMI-derived group velocities. Areas that contain fibrous tissues present the opposite findings. This leads us to understand that HU max could serve as a surrogate biomarker for the extent of collagen and likely its organization. Thus, we introduce the term breast compactness as a CT-measured surrogate biomarker for breast tissue collagen density, based on the maximum HU detected within a 3 millimeter region of interest. Breast compactness could aid breast cancer screening and risk assessment by pinpointing vulnerable breast regions, making it a promising imaging biomarker for breast stromal stiffness and early detection. Thank you for listening.
Thank you so much, Sofia. It's an amazing presentation. It's
Thank you.
Incredible how so many pathways and molecular processes are happening and we are not realizing it. And this is just the breast. It's not even the whole body. It's just unbelievable. And I have questions for you. So out of all of these molecular mechanisms that help or kind of lead to the accumulation of collagen in the breast, which one, in your opinion, do you think is the most critical for carcinogenesis and therefore should be watched in a more careful way?
Okay, I think I have 2 answers. I have a boring answer and a personal answer. The boring answer would be that FAK would be at the center of it all. And that's just because we know from data that it seems like it's the center of carcinogenesis, at least in these settings. In fact, we already know that FAK gene is amplified in about 40% of breast cancer specimens, but is minimal in benign breast epithelium. And then we know that FAK is associated with regulating a whole host of cellular activities. And so it would make sense that if it was to be altered, it would help carcinogenesis, and it would help cells survive in diverse environments and colonize new tissues. My more interesting or probably less correct answer would be that I think we should concentrate future research on the RHO FAK loop. And that's just because in our study, we saw that it was kind of the kickoff point for at least four new pathways that led to carcinogenic environments. So So those are my two answers.
Well, let me tell you, those two answers are both equally interesting and fascinating. So thank you so much. And I know for a fact that you dig very deeply in the literature out there to put all these mechanisms together, which is an amazing accomplishment. So congratulations for that.
Thanks.
And
it was hard because the more you dig, the more you find. And so at some point I just had to put a stop to it. Otherwise the paper would never have been finished.
But a good review is also the one that can also convey the information in a way that is easy to understand. And I think you excel on that. So that was great. And that takes me to the next question. So for all these molecular mechanisms, I'm just wondering, this is all happening in the breast, right? Where the primary tumor is. But when some cells travel in the bloodstream and they go somewhere else in the body, they probably, let's say they go to the liver or they go to the brain, they might find different microenvironments that are not necessarily the same as the one in the breast. So what do you think are the most important mechanisms that help these cells adapt to these new microenvironments so they can form new tumors?
Okay, so I think I already partially answered this in my boring answer to the first question. We know that FAK is so central, and we know that FAK has so many roles in just healthy cell life. And so we imagine, or we know from data, that if FAK is somehow impaired, that this helps cells thrive in new and diverse environments. So really FAK, I think, is the key to metastasization in this case. Yeah.
That sounds good. So we should definitely pay more attention to that particular pathway and molecule. Definitely. Yes. Thank you. And my last question is about the technology that you were talking about, the use of the measurements of Hounsfield units on chest CT as a way to predict the onset of breast cancer, and also to perform more accurate breast cancer screening. So if this materializes, if this method, the CT scan, becomes the standard of care to detect breast cancer, then in that future, where do you see the mammograms? Would the mammogram even play a role at all, or would this just be archived in the history of medicine?
Before I answer, I would say that I think we're very far away from being able to archive mammography altogether quite yet. We're not quite there yet. But I do think that a lot of data points towards the fact that CT outperforms mammography on a lot of tasks, if not most tasks that are useful to us in this realm of medicine. And so I see it as a very concrete possibility that CT could take the place of mammography. But I do see it as maybe towards the middle or end of my career, at least.
Well, hopefully sooner.
Yes, we'll see. We'll lay bets.
That's fascinating. Thank you so much. It's been an honor for me to share this space with you. And to our viewers, thank you so much for joining us. If you like what you learned today, please don't forget to subscribe and like this video and check out the prior videos that we have in our channel. And if you are an investigator and you wish to present your work here, please do not hesitate to reach out, and all the information will be available to you in the description box below. So thank you so much for joining us, and see you next time. Thank you, Sofia. Bye-bye.
Thank you. Bye.